Okay, chapter 31 is on plant structure, reproduction, and development. Alright, so there is a plant scientist, Natasha Rakel, who studies, I believe, at the University of California in Riverside, and she studies plants, and specifically the Arabidopsis plant, which is a type of mustard. And the Arabidopsis plant is a good plant to study because it has a very small genome. So it has very little DNA per cell, and I believe the genome has been entirely sequenced and all the proteins are known, which makes it a nice plant to use in the lab. The two main types of angiosperms are monocots and dicots, and that stands for monocotyledon, or one cotyledon, and dicotyledon, or two cotyledons. And you can see here the monocots have slightly different structures. So here's a monocot. It has literally one cotyledon in it. And when you talk about monocots, they have some things in common. The leaf ends tend to run parallel in the leaf. When we look at the vascular bundles, they tend to be, they say, in complex arrangements, but it's not really a nice circle like you will get in a dicot. You have some on the outside, and it'll almost look random on the inside under a microscope. If you take a cross-section and look at this, it kind of has a random look, but it's a very complex arrangement. The flowers will come in multiples of 3, and the roots are fibrous. So you can see they go everywhere. Grass is a nice example of this. The roots just kind of go everywhere versus the dicotyledon. The dicots have a nice taproot. All right, so here we go. Dicots have two cotyledons, obviously. The main vein is branched, so you can see it's not parallel. You still have one, and it comes into these branches. The vascular bundle has that nice ring. The floral parts are in 4 or 5, and again, they have that nice taproot. Okay, if we look at the body structure of a dicot, you have the terminal bud up here. And this is the bud that usually grows. There's apical dominance, which means that for a plant, the top part usually wants to grow. There are axillary buds, usually above each petiole and little leaf structure here. You can see one here and here. These will grow if you chop the top off. But usually with this apical dominance, the terminal bud grows first. You have, like I said, the leaf, you have the blade and the petiole, you have the stem, and in the stem, everywhere you have a leaf structure is called a node, and the part in between the leaf structure is called the internode. And then when we look at the root, we have this nice taproot, and we have all these little root hairs. And if you look over here, it's kind of cool. Each little root hair is actually just part of the epidermal cell that has been elongated. So that's actually one cell. You see one cell right here, and the root hair is just an elongated part of that. Okay, the plasma membrane of the root cell controls solute uptake. So in the soil there, they have to obviously take up not only water but minerals as well. And you can see that you have like tons and tons of surface area. So by having all these root hairs, you've greatly increased the surface area to uptake all that minerals and all that water. Okay, and many plants have modified root stems and leaves. And so these different modifications can do different things. If you modify part of the root system, we can store carbohydrates. And the plants obviously store carbohydrates for themselves. But us humans then like to dig them up and eat those stored carbohydrates, whether it's a carrot or a potato, anything that's sort of that tuber root, we like to dig up and eat. Okay, modified stems can do different things. Modified stems can be used for asexual reproduction, and the way that works is here's your strawberry plant, here's the mother strawberry plant. You see these? These are called runners typically, or stolen scientifically, but usually people use the term runner. That's a stem, and what it will do is that it will go into the ground and it'll produce a whole new plant, roots, leaves, everything, a whole new strawberry plant there. So that's asexual. These are obviously identical to the mother plant, genetically identical. You can modify the root structure by having something called a rhizome. A rhizome usually grows horizontal to the surface and stores carbohydrates. In this case, it ends in a very specific thing called a potato, which we again love to dig up and eat. Rhizomes are also things like ginger. So you can see here it's horizontal, and it's usually carbohydrate storage there. You can have modified leaves that are tendrils and wrap around things, or in the case of a cactus, you can have modified leaves that are prickly and help protect the plant. And also, by not having normal leaves, a cactus will lose less water. So a normal leaf tends to lose water fairly readily, and the modified leaf in a cactus will not. You can also modify a stem to act like a tendril, like a grapevine. The tendril there is actually a modified stem, not a modified leaf. Plant cells and tissues are diverse in structure and function. And so when we look at a typical plant cell, what we showed you in regular bio, and we call it a plant cell, that's what most cells have, but not all plant cells have the same thing. I mean, some of them don't even have chloroplasts, some don't have central vacuoles. Occasionally we get one that doesn't even have a nucleus, so they'll do different things, but the average one will have chloroplasts for photosynthesis, the central vacuole containing fluid, and remember that helps with the turgor, the pressure, pushing against the cell wall and giving that plant structure. And most of them will have a cell wall that surrounds the plasma membrane, and some of them even have a secondary cell wall. There are three types of tissue that make up a plant, and that's the dermal, the vascular, and the ground tissue system. And in this diagram, and pretty much I think every diagram in this lecture, the vascular system is in purple, so you can see here's a vascular bundle, as this is a cross-section of a stem vascular bundle. In this one, you can see it's in purple, right? The vascular bundles go down into the roots. The ground tissue is in like a yellow, and here that's the pith and the cortex. That's all ground tissue. If it's inside the vascular ring here, it's called a pith. If it's outside, it's called the cortex. And the dermal tissue, like the epidermis, the cuticle up here, that's in green. All right, and that's true in all the diagrams you're going to see. All right, so here's a cross section of a dicot and a monocot. And you can see they both have some similarities, right? They both have this nice epidermis, which is a really thick, not thick, I shouldn't say, really tight cells grouped together very tightly, and it tends to make a cuticle to really protect the plant. They both have vascular bundles, but here in the dicot, we have that nice ring. All right, there's the ring with the xylem and the phloem. Xylem, if you remember, carry the water. Phloem carry the carbohydrates. So xylem, the water goes up the plant, and phloem is usually carrying carbohydrates down. So here's your vascular system in a monocot. Again, you have a ring, and they said it's complex, but it's actually so complex it'll look random as you look at it. That's in a monocot. They both have ground tissue. In the dicot, again, the pith is inside the vascular ring, and the cortex is outside. Both of them still ground tissue, though. All right, there are 5 major types of cells. The parenchyma, which performs most of the metabolic functions. And when we talk about cells, we're usually talking about the parenchyma. The most cells that you look at are the parenchyma. The collenchyma provide support for the growing cells. So when we look at Sclerenchyma, I think a good example of this is celery, the stringy part. They provide support for the growing part, not the dead part of a plant, but the part that's still growing. Another kind of plant cell that there is something called the Sclerenchyma, and that's the main component of wood. On the left, you're going to see a fiber cell, and when those die, they become the skeleton of the tree. And you can see in this slide here the fiber cells. On the right, you have something called a sclereid, and these are really tough. They have a very thick but irregular secondary cell wall, and they make up nutshells and the gritty part of pears, and they are very, very tough. So primary growth lengthens the roots and the shoots. It makes it taller and makes the roots go deeper. Most plants exhibit something called indeterminate growth, and that means they continue to grow and grow and grow as long as they live. That is unlike animals or humans. We grow to a certain size and we stop, and that is called determinate growth. We grow and then we reach a size and we stop. We don't get bigger and bigger and bigger forever. Okay, the apical meristems are located in the tips of the roots and the terminal and axillary buds of shoots. And these are going to do the primary growth. So you can see the terminal bud is up here, and this is the bud that's going to do the growing. Unless it gets cut off, this is going to have the apical dominance at the top here. And down here you have the roots that will grow for the primary growth. All right, in a root tip, what you have is something called an apical meristem region, and that is down here. And if you think about these cells, these are cells that are growing and dividing and immature, and you can't just shove them through the soil. So there's something called a root cap, and that protects the cells as the root grows and elongates. And that root cap will protect those dividing cells from the soil. All right, the axillary bud meristem are found near the apical meristem. So here you go, it's just on either side. And when you look at this diagram, this dark purple staining is where you have cell division, rapid cell division. So you have it here at the apical meristem, and you have it here. Secondary growth is different than the primary growth. Primary growth is going to be increasing the height and the depth of the roots. Secondary growth is going to increase the girth of the woody plants. It's going to make them wider. Okay? Secondary growth arises from cell division, and it arises in a specific location. There is a cylindrical meristem called the vascular cambrium. All right, the vascular cambrium is in this diagram right here. You can see this blue line. That's the vascular cambrium. And on the left of it, you have your primary xylem, and on the right, your primary phloem. And you can see this growth chart. It's pushing these arrows, say that it's going to grow from the vascular cambrium. It's going to keep putting down primary xylem on its left. It's going to put down phloem on its right, and it's going to push this out from this location. So this is in year 1, or the early spring. And here you go, you can see the secondary xylem being laid down here. It's on either side of the vascular cambrium. And it's going to continue to push this out. Now the epidermis here gets shed at approximately the same rate it gets created. All right, so here we have the heartwood, and you can see these nice growth rings. And the growth rings are created, especially in temperate areas, by, in spring you have a lot of rain, and the xylem's put down very quickly, and the cells tend to be thin. And then in summer, things tend to dry up, and what you get is a very nice rings for each year. And inside of this heartwood, this is all dead. This xylem and phloem is no longer working. It's actually filled with a resin that helps give structure to the tree. The xylem and phloem out here in the sapwood, it is what is keeping the tree alive. All right, annuals complete their life cycle in a single year. That's the definition of an annual. There are examples of that: wheat, corn, rice, wildflowers. Biannuals complete their life cycle in 2 years. They flower in the second year. Examples are beets and carrots, although when those are used in agriculture and they're harvested, they're treated more like an annual. And then perennials, what I like to plant in my garden because I don't like to replant every year, those live and reproduce for many, many years, and it depends on the species how long they live, but they live essentially for many years until they die, 7, 10, 100, and those are perennials. Okay, so for reproductive plants, and we're looking at angiosperms, we're looking at flowers here, you're going to have two sides of it. The carpel is going to be the female side. You have the ovary here, you have the style and the stigma here. The stamen over here with the anther and filament is the male side. This is going to produce the pollen over here. All right, so for flower reproduction, you have the tip of the carpel up here, and the stigma up here receives the pollen grain. The ovary at the base of the carpel is where the egg is, and the ovule will turn into the seed. The ovary is what's going to turn into the fruit. And then this just shows here, if you follow it around, the life cycle where you have the seed, a germinating seedling, and back to a mature plant. The gametophyte development and fertilization in an angiosperm, I really suggest you look this diagram up in the book and study it a bit because it gets a little complicated. What you have is the anther here. You're going to have meiosis, all right, and we're going to produce the 4 haploid spores. We're going to have mitosis that's going to produce the pollen grain, and we're going to have 2 cells produced. Those are going to pollinate here, and this is actually 2 sperm in the pollen tube. You actually end up having 2 fertilize this, which is interesting. Up here you have the ovary and the ovule. Meiosis, again, to produce the egg and the embryo sac. So that is also produced by meiosis, right? So what happens with this double fertilization is one sperm is going to help produce this endosperm, and it becomes a triploid endosperm. And you think triploid's kind of weird, it's a 3-end situation, but that is just going to be used to house and provide food for the seed. It's not going to reproduce again. What you end up down here with is the diploid zygote. This is the one that's going to produce the actual plant. Okay, the ovule here is going to develop into the seed, and you can see it. You can see it over right here. After fertilization, it becomes a seed, and again, the ovary will become the fruit. You can see here, in this case, it is a dicot. You can see 2 cotyledons. You can see the seed coat and the endosperm right here. You can also see the root. Okay. Here is a nice picture where you have a dicot and a monocot. A bean is a very classic example of a dicot. I don't know if you've ever done the science experiment where you put beans in a Ziploc bag with a little wet Scott towel, but you clearly see the root coming out. This here is the embryonic leaves that are in there and the seed coat. So, and each side of these is a cotyledon. When you cut a bean in half, you're looking at 2 cotyledons. A monocot, a little different. You have the fruit tissue here on the outside and the seed coat, but look inside here, you have something a little different. You have this sheath. You have the embryonic leaf, which is similar to a dicot. and then you have the shoot and the root here. Okay. You can get fruits that have one flower but a bunch of carpels, and they become an aggregate fruit, and a pineapple is a nice example of that. And it's basically many different fruits that grow together. There's one flower but a bunch of different carpels. They all get fertilized, and you really essentially get an aggregate fruit or a bunch of them that end up growing together. Seed dormancy is an important characteristic in which the growth and development are suspended temporarily. And the reason that plants will do that is, one, it'll help disperse the seed. So whether the seed is being dispersed by the wind or whether an animal is carrying it in a fruit, whatever, however it's being dispersed, it gives it time. But the other thing, the really important thing, is it will increase the chance of having good environmental conditions for survival. So essentially it can wait until it rains, there's water, and then the seed can germinate. In a dicot, this little diagram here is nice. You can see in a dicot the root emerges first, followed by the shoot, which is covered by that protective hook. Monocot, a little different. There's the protective sheath surrounding the shoot, and that's what breaks the soil. Then you get, again, the foliage coming out second. Asexual reproduction produces plant clones, and that means they are genetically identical to the mother plant. And you can do it in different ways. You can have the garlic, which is a bulb. You can have sprouts off of the side. You can have runners. In this case, in this desert plant, this ring here, these are clones of the mother plant, and this one I think they think is about 12,000 years old. The ring just increases in diameter as these runners go out in this desert plant. Okay. Asexual reproduction is a mainstay of modern agriculture. So they propagate plants from cuttings or bits of tissue, and that is good for agriculture. When we talk about sexual reproduction, we say, well, it increases variety, and that's good for evolution. But when you're looking at agriculture, you don't want variety. You want to know that whatever plants you have planted have the characteristics you have worked for. And the way to reduce variety is through asexual reproduction.